Hi, welcome to the Introduction to Genetics podcast. We're going to start with talking about an individual named Gregor Mendel who lived in 1822 and died in 1884. And his research is often called Mendelian genetics. So, who was Gregor Mendel? He is, if I can move this recording screen, this fella up in the right hand corner, he was a monk in Czechoslovakia. And he was a priest, actually, and he was the son of peasants, and he was really much interested in how traits were inherited and how parents passed on traits to their offspring. And so he spent a lot of time working with pea plants in the garden, um, crossing them and growing them and seeing what traits he could get. And so he carried out these experiments with various traits of pea plants. And over to the right here is just an example of a trait that he studied. He crossed a pea plant that produced smooth seeds with a pea plant that produced wrinkled seeds. And you can see this sort of equation up here. And you'll get used to doing quite a few equations here um, in this genetics unit. You see the smooth parent and the wrinkled parent, and he ended up getting a smooth parent, and then he used the plant to self-fertilize, and he got in the F2 generation, that second generation, a smooth plant and a wrinkled plant. And he said, well, how could that be? Because when I first initially crossed these two, I crossed a smooth and a wrinkled and got a smooth. And when I crossed them again, I got a smooth and a wrinkled. So he was naturally very curious as to what was going on and what was happening, what parents were passing on what traits. So if we look at the parent generation, and again, this is right in your notes, just make sure you make note of that. When he was studying this, he had the smooth and the wrinkled. They're called the P generation or the parent generation. The F1 generation are the offspring, and all of them in this case were smooth. And this showed Mendel's law of dominance. And that just states that instead of blending together, some traits will override other traits. And so right here we see that smooth trait overriding the wrinkled traits. And then he used, of course, that F1 generation to self-fertilize and create the F2 generation, the offspring of the offspring. And he found out that about 75% were smooth and about 25% were wrinkled. And so what he realized in this one, because he found in the offspring of the offspring, that F2 generation, that the wrinkled version of this trait showed up, and that he deemed these as recessive traits. And these are the traits that are expressed only if a dominant trait is not present. And so again, you look back at the parents and you have the smooth trait, which is the dominant trait, and the wrinkled trait, which is the recessive trait. And uh, having them have offspring and the offspring have offspring, that recessive trait was just hidden until the correct opportunity, the right opportunity showed up, where you had both traits being recessive, giving that phenotype or that physical characteristic of a wrinkled seed. So what did he conclude? He concluded that genes code for traits like seed shape or eye color or hair color. Those are certain traits, and that alleles are different versions of a gene. So smooth or wrinkled or curly hair, or wavy hair, blue eyes or green eyes, or tall or short. Those are all different versions of the gene, and we call those alleles. And that he also noticed that some alleles are dominant and some are recessive, and that each organism contains two alleles for each trait. Alleles separate, and again, going back to things like meiosis and mitosis, we're going to jump back all the way there, but alleles do separate during the formation of sex cells, and that's all about meiosis, and that's what we learned about a few a few units ago, and that's known as the law of segregation. And this is what brings up a lot of diversity. The alleles separate during the formation of sex cells, and which way they separate is completely random. And so that's what he concluded from doing all these P plans. And he really set the bar for the rest of the science world when it came to studying genetics. So we can thank him for the Human Genome Project, which was completed not too long ago, a little over 10 years ago, that mapped out all of the human genes. And he kind of started this whole process of just thinking about traits and where they came from, and alleles, and dominance, and recessive, and all these terms that we find in genetics today. So that does it with this introduction to genetics.